My name is Lizzie Chung, and this summer I'm working on characterization of human macrophage polarization with the Laufenberger lab. So, for some quick context, macrophages are immune cells involved in the detection, phagocytosis, and killing of pathogens and disease cells. And macrophage polarization refers to how macrophages adopt different functions in response to changes in their environment. Historically, macrophages have been categorized according to the M1 and M2 phenotypes, whereby M1 represents a pro-inflammatory activity and M2 represents anti-inflammatory activity. However, thinking has recently started to shift towards this idea that macrophage polarization actually lies on a spectrum between M1 and M2 states. And there's a need for us to better understand this because macrophage polarization state can impact the disease environment and disease progression, especially in inflammatory conditions. So my goal this summer was to work on developing an in vitro system to study macrophage polarization. The goals of this are one, to better characterize macrophage polarization as a function of protein secretion, and two, to determine how macrophage polarization state may contribute to disease such as tumor growth or viral infection. So a general schematic of our project and how we obtained this was by going to Massachusetts General Hospital to get Buffy coats, which are blood samples that are centrifuged to have a more concentrated solution of white blood cells and peripheral blood mononuclear cells. And from this, we isolated monocytes, which we further differentiated into macrophages. And after differentiating them, we treated them with different conditions to polarize them into the M1 and M2 states, respectively. So we chose our treatments based on literature reports of common macrophage stimuli and our TLR agonist based on experimental interests. And these are implicated in the targeting of Epstein-Barr virus. We were able to run Luminex and ELISA for assaying protein secretion across different treatments. And we also did cell imaging for looking at morphology and quantifying CD86 receptor expression. Cell imaging reveals morphology profiles consistent with literature, whereby M1 macrophages display a stretched and elongated phenotype, and M2 macrophages display a large, round, and flat morphology. We ran a Whitney-Mann test on our fluorescence where DAPI in blue stains our cell nuclei and GFP stains CD86 receptors. CD86 receptors are involved in T cell regulation, and so visually we see an increased GFP expression in interferon gamma, which implies upregulation of CD86 and which further indicates that there's an inflammatory response occurring. In this, we see between the median IL-4 and median interferon gamma conditions, a p-value that's not significant, but between IL-4 and interferon gamma, we see a p-value of 0.06, which is close to the cutoff for the 0.05, indicating that we may see some sort of upregulation in interferon gamma. Our ELISA demonstrates an upregulation of CXCL10 in inflammatory conditions. To the left, I have the standard curve, which is on a log scale and referring to picograms per mil. And then our bar plot demonstrates upregulation of CXCL10 across the different treatments, but a higher levels of it with interferon gamma and interferon gamma with lipopolysaccharide, which makes sense considering that it is involved in inflammatory conditions and interferon gamma is also implicated in inflammation. Our Luminex data shows us that macrophages display distinct protein signatures in different environments, whereby we see interferon gamma leading to high levels of cytokine production. And then our PCA analysis shows separation across different conditions and clustering within the same condition, where each dot on the plot represents a different replicate of the condition referenced based on color. And so based on this, it's confirmed that we do have differential protein secretion and that polarization state will influence the amount of cytokines and the total immune system response. So in conclusion, macrophages are highly plastic and exhibit differential protein secretion depending on disease environment. CXCL10 secretion is upregulated in inflammatory conditions. Macrophages treated with interferon gamma display increased expression of CD86, and interferon gamma induces pro-inflammatory cytokine secretion by macrophages. I'd like to thank everybody who contributed to helping me on this project, especially Krista Pullen and Anisha Datta, and then also to Dr. Doug Laufenberger for allowing me to work with his lab this summer. Thank you.